All right, welcome back to the deep dive. Today we're taking a look at, well, I'd say a crucial step in drug development, data analysis in those super early clinical trials, phase 1 and 2. Where a potential new treatment, it gets tested in humans for the very first time.
Exactly. These early studies, they're all about gathering that initial information on safety, figuring out, is this drug safe for people to take? And do we see any early signs, any hints at all that, it might actually work?
And just so everyone's on the same page, what we want to do today, our goal, is to really dig into those fundamental statistical methods, the nuts and bolts, of how researchers interpret the initial signals, the early data coming out of these trials.
Right, because it's not always straightforward. We'll also be discussing how researchers navigate those tricky decision-making processes, based on those initial findings. And of course, we can't forget about variability, those inherent differences in how people respond to a drug. We'll be exploring how that plays a role in the analysis.
And of course, the FDA, the ICH, all those regulatory guidelines will come into play. And speaking of sources, we've got a lot to work with today, actual drug discovery case studies, the basic principles of medicinal chemistry, like how drugs are designed, some overviews of the whole drug development process. Then we've got stuff on preclinical testing, clinical trial design, and then, of course, we have those essential regulatory guidelines.
Yeah, real smorgasbord. And all of these different perspectives, they're going to give us a pretty comprehensive picture, of the challenges and considerations in analyzing data from those early human studies.
So to kick things off, let's really break down what data analysis actually looks like in these initial phase 1 trials. What's the main aim?
Yeah.
What are we trying to achieve?
Well, in phase 1, the data analysis, it's laser focused on safety and tolerability.
Yeah.
We're talking about meticulously collecting information on any adverse events, any side effects the participants experience.
Right. So we're watching very carefully for any potential red flags.
Exactly. But at the same time, we're also gathering those crucial early insights into pharmacokinetics. Basically, we want to understand how the drug moves through the body, how it's absorbed, distributed, metabolized, and finally eliminated, the whole journey.
So it's like we're creating a map of the drug's adventure through the body. But phase 1 trials are usually pretty small, right?
Mm-hmm.
Limited number of participants, often healthy volunteers. How does that impact the way we can analyze the data from a statistical point of view?
Yeah, that's a really important point. With smaller numbers, we have what we call lower statistical power. That basically means it's harder to detect those subtle, maybe less obvious safety signals simply because we have fewer data points to work with. So our interpretations at this stage, they have to be very cautious, taking into account those limitations.
So it's all about nuance, really
Okay.
considering the context.
Absolutely.
Okay, so let's dive a little deeper into the safety data itself. How are these adverse events actually collected and analyzed? What are researchers really looking for in all that information?
So when an adverse event happens, it's documented very carefully. We're talking about recording what happened, how severe it was, mild, moderate, severe, how long it lasted, and whether it could potentially be related to the drug. And then we use descriptive statistics, a fancy way of saying we're looking at things like, how often different types of adverse events occur.
Right, so like what percentage of people experience a particular side effect?
Exactly. But a really critical goal here is to identify those dose-limiting toxicities, those DLTs. These are the serious adverse events that happen at a specific dose, and they basically prevent us from safely increasing the dose any further.
So they're like, those warning signs telling us, hey, this is where it starts to get risky.
Yeah, exactly. Analyzing when and at what dose these DLTs pop up, that's fundamental to figuring out what's called the maximum tolerated dose, the MTD. It's that delicate balance, right? We want to find that sweet spot where we're getting the potential benefit of the drug without pushing people into dangerous territory.
It's all about finding that fine line between benefit and risk. Now, you mentioned pharmacokinetic data earlier, that whole journey of the drug through the body. What's the analysis process for that? I remember hearing about something called plasma concentration curves.
Right. So pharmacokinetic data, it's all about tracking the drug's concentration in the body over time. So imagine, someone gets a single dose of a drug intravenously, like a rapid injection, what we call a bolus dose. We can then measure how much drug is present in their blood plasma at different time points, like 30 minutes later, an hour later, 2 hours later.
So we're basically taking snapshots of the drug's levels at various points along its journey.
Exactly. And if we plot those measurements on a graph, We get what's called a plasma concentration versus time curve. You can think of it as visually mapping out how the drug travels through the body.
So that's like a visual representation of that journey.
Right. And then we use these things called pharmacokinetic models, these mathematical equations that help us make sense of those curves. And from there, we can estimate those key parameters, like how long the drug stays in different parts of the body or how quickly it's eliminated.
So we're not just seeing the drug's movement, we're actually understanding the mechanics of that journey.
Exactly. And in relation to those curves, you mentioned MEC and MTC. So MEC stands for minimum effective concentration, and it's basically the lowest concentration of the drug needed to actually have a therapeutic effect.
Right, so we need to hit at least that level for the drug to do its job.
Exactly. And MTC, on the other hand, stands for minimum toxic concentration. That's the level above which we start to see those unwanted side effects. So in drug development, we want to keep that drug concentration within what we call a therapeutic window. It needs to be of the MEC so it's effective, but below the MTC to avoid toxicity.
So like a Goldilocks zone for drug levels.
Yeah.
Not too low, not too high, just right.
Exactly. These pharmacokinetic parameters are crucial for figuring out that perfect zone.
Okay, so we're gathering all this data on safety and pharmacokinetics in phase 1. How does this information actually guide the decisions about moving forward with the trial, especially when it comes to deciding whether to increase the dose?
Yeah, that's a really important part of the process. So dose escalation in phase 1, it's a very carefully controlled process. We usually have these predefined rules outlined in the study protocol, and those rules help us make those decisions based on the safety data. For example, if we see too many DLTs at a particular dose, we know we can't go higher. We might pause the trial or even reduce the dose to ensure patient safety.
It's like that cautious climb up a ladder. We take one step at a time, checking for stability before moving higher.
Exactly. And there are some early statistical models that we use to help with these decisions. We have something called up and down designs, which guide us in finding that maximum tolerated dose efficiently, even with limited data. Another one is the continual reassessment method, or CRM. It's more complex, but it allows us to incorporate more information as we go along.
So it's like those models, they act as our guide as we carefully navigate this dose escalation process.
Now, you mentioned earlier about variability in this early human data. How much of an impact does that have on the analysis, and how do researchers take that into account?
Right, variability in phase 1 data, it can be pretty significant. I mean, people, they metabolize drugs differently, they have different body weights, different underlying health conditions. So statistical analyses, they need to consider those individual differences. We can't just focus on the average response. We also need to look at the range and spread of the data to get a more realistic picture.
So it's about appreciating those individual responses, not just looking at the big group average.
Exactly. Even with a small sample size, we want to understand how the drug might behave across a more diverse population.
Okay, so phase 1, we're figuring out safety, we're understanding how the drug behaves in the body. Now, let's switch gears to phase 2. What's the main goal of data analysis at this stage? So in phase 2, safety is still paramount, of course,
Yeah.
but the focus really expands to include efficacy. We want to know, does the drug actually work? Does it show signs of having a beneficial effect in patients who have the specific condition we're targeting?
So it's like now we're really starting to ask, can this drug deliver on its promise?
Exactly. And this is where analyzing what we call efficacy endpoints, those measures of how well the treatment works, becomes really central to the whole data analysis process.
And phase 2 usually involves a larger group of participants than phase 1, right? What kind of impact does that have on the analysis?
Yeah, phase 2 trials typically involve a bigger group, which gives us more statistical power. We basically have a better chance of detecting a real treatment effect, if the drug is actually doing something. And it also allows for more robust, more reliable analyses of those efficacy endpoints.
So a bigger sample size helps us see those effects more clearly, even if they're subtle.
Precisely.
Okay, let's talk about those efficacy endpoints. What kind of data are we actually collecting, and how is it analyzed?
Well, the types of endpoints, they can vary quite a bit depending on the condition being studied. So, for instance, in cancer trials, we might look at tumor shrinkage, a continuous variable, as the primary endpoint. In hypertension, we might look at changes in blood pressure. Sometimes those endpoints are binary, like did the patient respond to treatment or not, a yes or no type of outcome.
So different diseases, different ways of measuring how well the treatment is working.
Right. And the analysis methods, they also differ. For those continuous endpoints, we often compare the average change in the group getting the drug versus a control group, folks who might be getting a placebo or the standard treatment. And with those binary endpoints, we might compare the proportion of responders in each group.
And this brings us to the whole idea of a control group, right? Why is having that comparison group so critical for analyzing efficacy?
Absolutely. The control group is the key. Without it, we can't be sure if the changes we're seeing are because of the drug or because of something else entirely, like, the disease just naturally getting better or the placebo effect. Randomly assigning people to either the treatment group or the control group, that helps us minimize bias and make a fair comparison.
So it's like we're leveling the playing field at the start, making sure the groups are as similar as possible except for whether they get the drug or not.
Exactly. And then we use these things called inferential statistics, which help us decide whether those differences between the groups are likely real or just due to random chance.
Right, so we want to be confident that the drug is actually making a difference.
Exactly. We're looking for statistical significance, which basically means the results are unlikely to have happened by chance alone.
Now, I've also read about these things called response rates, especially in cancer trials. Can you explain how those are analyzed?
Yeah, so in phase 2 oncology trials, the response rate is a big one. It's the percentage of patients whose tumors shrink by a certain amount after treatment. And to analyze those response rates, we calculate the observed response rate in the group getting the drug and often create a confidence interval around that estimate. It basically gives us a range of values where the true response rate in a larger population likely falls.
So it's like we're trying to understand how reliable our estimate is.
Right. It gives us a sense of the precision of those findings.
Now, phase 2 trials, they can run for a while, and I understand there are times when researchers can actually peek at the data before the trial is officially over. These are called interim analyses, right? What's the
Yeah.
point of doing that?
Yes, exactly. Interim analyses are like these planned check-ins along the way. And the main reason we do them is to look at the data early on and make informed decisions about the future of the trial. So, for instance, if the early data are really promising, we're seeing strong signs of efficacy, we might even stop the trial early.
So it's like we don't want to keep people on a placebo or an inferior treatment if we already have good evidence that the new drug is working well.
Exactly. On the flip side, if the early data show that the drug isn't working at all, we might stop the trial for futility. We don't want to continue giving a treatment that's unlikely to benefit anyone.
So those interim analyses, they're like those critical decision points that can change the course of the trial.
Right. They're important for both ethical and practical reasons.
I'm guessing those decisions to stop or continue the trial, they're not just made randomly, right? There must be some kind of guidelines in place.
Oh, absolutely. We have these predefined statistical decision rules that are laid out in the trial protocol before the trial even begins. These rules clearly define what criteria would trigger a decision to continue, to stop for futility, or to stop early for those really promising or concerning results. Sticking to these rules is crucial, to maintain the integrity of the trial and to make sure our decisions are based on solid data.
So it's like having a clear roadmap that keeps everyone on the same page and helps ensure we're making sound judgments.
Exactly.
Now, we talked about variability earlier in the context of phase 1. Is that still something that researchers need to consider when they're analyzing the data from phase 2?
Absolutely. Even though we have a larger sample size in phase 2, people are still different. They can respond to the same drug in different ways due to their individual characteristics, their genetics, their overall health, and so on.
Right, so those individual differences are still in play.
Exactly. And we need to account for that variability when we're interpreting the phase 2 data. Some statistical models actually include ways to estimate and adjust for those patient-to-patient differences.
So it's like we're acknowledging that not everyone is going to fit neatly into the average.
Precisely.
We've discussed the types of data and some of the statistical concepts. Are there specific statistical models that are commonly used in these early-stage clinical trials?
Yeah, there are some go-to models, although we usually try to keep things relatively simple at this stage. So for continuous endpoints, like measuring a change in blood pressure, we might use a t-test. That helps us see if the average change in the treatment group is truly different from the average change in the control group. Or we might use something called ANOVA if we have more than two groups to compare.
Right, so it's like choosing the right tool for the job based on the type of data we have.
Exactly. Then, for binary endpoints, those yes or no outcomes, we often use logistic regression. That helps us figure out the odds of a particular outcome happening in different groups, like the odds of responding to treatment.
So we're not just looking at whether there's a difference, but also trying to quantify how likely that difference is.
Precisely. And sometimes our endpoint might be time to an event, like how long does it take for a patient's disease to progress. In those cases, we might use survival analysis techniques. We can create these Kaplan-Meier curves, which visually show how the time to event differs between groups. And we can use Cox proportional hazards models, which help us statistically compare those time to event differences while accounting for other factors that might be influencing the results.
So a whole range of models to help us make sense of different types of data.
Right. And the specific model we choose really depends on the research question we're asking and the characteristics of our data.
Okay, so we run these analyses and we get our numbers. But how do researchers actually interpret those results? What does it all mean in a practical sense?
Right, interpretation. It goes beyond just looking at whether a result is statistically significant, whether it's unlikely to have happened by chance. We also need to consider what's called clinical significance.
So is it meaningful for actual patients?
Exactly. It's not just about numbers, it's about impact. So even if a result is statistically significant, the effect size, meaning how much better the drug performed compared to the control, that has to be big enough to actually make a real difference in people's lives.
Right. A small improvement might be statistically significant, but not really worth it from a patient's perspective.
Exactly. And we also have to look at the safety data alongside the efficacy data. A drug that shows a statistically significant benefit but has really bad side effects, well, that might not be a good candidate for further development.
So it's like a balancing act, weighing the potential benefits against the potential risks.
Precisely. And this is where clinical judgment really comes in.
Now, we've been focusing on the scientific aspects, but what about the regulatory side of things? How do agencies like the FDA and ICH view all this data analysis in these early trials?
So regulatory agencies, their primary concern in these early phases is safety, first and foremost. They want to make sure that trial participants are protected and that the risks they're exposed to are justifiable. So they want to see a very thorough and rigorous analysis of all the safety data. They have very specific guidelines that dictate how we need to collect, analyze, and report that data.
And what about in terms of efficacy? Do they have expectations for phase 2 data?
Yeah, for a drug to be considered truly effective by the FDA in those later stages, for it to get approved, we usually need to show statistically significant and clinically meaningful results on those pre-specified primary endpoints that we outlined in the trial protocol.
So it's all about setting those goals up front and then demonstrating that the drug can hit those targets.
Exactly, and there are some key guidelines that they refer to. For instance, there's ICH E6, which provides comprehensive guidance on good clinical practice, or GCP, which covers all aspects of clinical trial conduct, including making sure the data we collect is high quality and reliable. And then there's ICH E9, which specifically focuses on statistical principles for clinical trials. So it's all about making sure that we're using sound statistical methods throughout the trial, even in these early stages.
Right, so those guidelines provide a framework for conducting those analyses in a rigorous and trustworthy way.
Exactly.
And what about the Investigational New Drug application, the IND? I remember that being a big step in the drug development process.
Yeah.
How does the data analysis from those early trials play into that?
So the IND is basically the sponsor's request to the FDA to allow them to move into those later stage trials. And the data from phase 1 and 2, that forms a critical part of that application. The FDA reviews all that data very carefully to assess the drug's safety profile and to decide if there's enough evidence of potential benefit to justify further investigation. A strong, well-analyzed, early-stage data set that's really key for getting the green light to move forward.
So it's like passing a crucial checkpoint on that long road to drug approval.
Precisely.
Wow, this has been a really fascinating deep dive. It's clear that data analysis in those early stage trials, it's so much more than just crunching numbers. It's a complex process with a lot of careful consideration and interpretation involved.
Absolutely. It's about bringing together statistical rigor, clinical judgment, and a deep understanding of those regulatory requirements. And it's all in service of making sure we're developing new therapies in a way that's safe, ethical, and ultimately beneficial to patients.
So as we wrap up, here's something for everyone listening to ponder. We've seen how much uncertainty there is in these early phases. We're working with limited data trying to piece together those early signals. So how do researchers and regulators, how do they balance that need for solid evidence with the very real urgency of developing treatments for diseases where, there might not be any good options right now?
It's a challenging question, and it's one that really shapes the whole landscape of drug development. Thanks for joining us.